Meet Claudia, a computational biologist who used Claude Science to analyze the primary human CD4 and T-cell Perturb-seq dataset but wants to audit the claims. That's why I built GeneGround, a claim-level grounding layer for AI-generated omics interpretations. It ingests interpretation in a zip file with compact evidence packets like differential expression evidence and pathway signatures. Behind the scenes, Claude first extracts individual biological claims from the interpretation. Then deterministic code normalizes these claims against ontology, so genes are mapped to HGNC symbols, pathways to the reactome database or curated immune signatures, and cell types to the OBO Foundry cell ontology. The Claude Science handoff file is parsed into 5 evidence indexes like provenance or language rules, where GeneGround then retrieves small evidence chunks using metadata matching and TF-IDF lexical vector search as a fallback, comparing agent queries against embedding-ready chunk summaries. Finally, each claim is evaluated against these evidence chunks by 4 Claude agents, including perturbation evidence, pathway signature, robustness quality, and language causality, and the final verdict is assigned by deterministic aggregation rules. The result is a rewritten interpretation with a claim-level audit. So in this case, 8 claims were extracted, with 6 supported with caveats, 1 partially supported, and 1 overstated. This NFKB2 claim is supported with caveats because the on-target effect size and differential expression gene counts are supported, but the transcriptional response is correlational and not direct proof. This claim on the distinct arms of the CD4 and T-cell response is overstated because the evidence supports transcriptional themes but not a fully defined response architecture. The user can select a phrase and ask a question or propose a change to the review system powered by Claude API. If a proposed rewrite is approved, GeneGround applies the change directly to the interpretation. The technical pipeline makes the process transparent. You can click a claim to view its status and reasoning. This page shows the input source, extracted claims, the number of evidence chunks in each evidence index, the number of retrieved chunks per claim, and any warnings or fallbacks. Clicking into more details shows the chunks actually reviewed for agent evaluation, and all the evidence indexed from the user's inputted file. And then clicking Show Metadata shows the details like the original source file and artifact, dataset, and target gene symbol, making the system auditable instead of black box. Finally, the evidence trace links every claim to its corresponding evidence, categorized by agent and retrieval status, including metadata exact match, partial match, or lexical vector search. This statement features evidence from the differential expression H5AD file and pathway enrichment results CSV. So overall, with the development of Claude Science and other AI tools to expedite omics analysis, GeneGround's goal is to make that AI-assisted science more transparent. Thank you so much for watching.